So, good morning, everyone. For this session, the speakers will have 5 minutes to show us projects and initiatives that are important to Latin America bioimaging community. We will have maybe 2 minutes to make questions or comments, so I would like to encourage you all to contact them directly because every session is networking session if you will want. So to begin, I would like to welcome Charlotte Buckley from Universidade Maior Santiago, Chile.
Hi everyone, it's really great to be here today. I recently moved from Scotland to Chile, so this is such a fantastic introduction to the vibrant Latin American bioimaging community. So this one? Sorry. Today I'm going to talk to you about some core techniques that I use in my research, which is calcium imaging and signal analysis. So I use this to try and understand intercellular signaling in blood vessels. So some of the main ways that cells release calcium from the internal stores via IP3 receptor binding of IP3. So the ways that I can experimentally stimulate this are to either use an external agonist, which binds to, will bind to a GPCR on the surface of the cell and cause PLC to activate the breakdown of PIP2 into IP3. The other way that I can use is to use a caged compound, so a caged form of IP3. This is inactive until you shine UV light on cells where this compound has been incubated. And then you get release of the caging group, allowing you to really precisely release calcium in specific cells or within that cell. I image the calcium using Cal520. So I work with endothelial cells, and we really like the Cal520 indicator dye. And I use this either on freshly dissected primary arteries, which I dissect out and pin open and stain, or on primary endothelial cell patches, as you can see on the bottom here. The imaging systems that I use are fairly simple, actually. For the arterial preparations, I use an upright microscope, and for the cells, I use the inverted system. For each of these, we use a high-sensitivity EMC-CD camera and LED illumination, and we like to keep the light levels really low, so often with neutral density filters in the way to keep the light levels very low. We 3D print, design and 3D print sample holders. This is so that we can get nice laminar flow across the preparations, and also bring in micromanipulators and pipettes so that we can apply agonists in very specific locations on the preparations. And we use UV illumination to actually cause the photolysis. So these are wrap electronic systems. We use a laser illumination system, which allows you to really specifically choose the cells you want to shine the UV light on. Or here with the flash photolysis system, which is incredibly quick within a single 10 millisecond frame. So what can I do with these tools? So these are some of the different types of imaging that we can do. With the UV photolysis, which occurs within the central region here, you can see that once we get this uncaging, you get this beautiful calcium wave propagating out from the uncaging region. We can also specifically increase calcium in an IP3 independent manner here with MPEGTA, and then buffer calcium. So here I'm using the diazo-2 AM dye. Similarly, with agonist perfusion, here I'm applying acetylcholine, which is stimulating this IP3 generation within the cells. Always add in sulforhodamine to be able to tell exactly which cells the agonist is interacting with. An IP3 independent way of doing it is by putting on ionophores like ionomycin in this central video here. And then it can also buffer calcium with BAPTA, as you can see here. So lots of different ways, a quick overview of the different ways that we can actually stimulate calcium release and buffer. So to analyze this, we use a mixture of manual and automated analysis techniques. So manually labeling the cells, because we haven't been able to actually get any really good automated way of finding cell boundaries. So if anybody knows how to do that, please come chat to me. And then these locations and all of the stacks are loaded into a home-written Python script where we find the raw signals, identify the baseline, and find the f/f naught signals. To then identify the peaks, we take the derivative, and then with these peaks, we're able to identify active cells and extract loads of different parameters, such as the peak signal, time of signal, different number of oscillations. And then with these, we can then find the averages across different biological replicates. So with these techniques, we can look at thousands of cells from the same preparation and analyze the signals really rapidly. And I would say that this, all of this software is being put together into with a nice user interface. It's going to be published soon and be open source. So that's called CalTracker, so look out for that soon. And if anyone's interested in any of these techniques, please come see me at my poster. Thanks very much.
Thank you so much. Some comments or questions?
Thank you, Charlotte, for your amazing presentation. I would like to ask you if, considering the speed limit of your technique, if you can use this to analyze, for example, in cardiomyocytes and other, electrostimulate cells, if you have tested this. Thank you.
I haven't tested it, but yeah, I think that would be very doable, yeah.
Okay.
Yeah, I'd be happy to chat to you later about it.
Someone else?
Thank you, awesome presentation. I would like to know what kind of fluorescent proofs are you using? Because I'm also analyzing calcium currents, but we're contending with the photophysic property of the fluorescent proof about the making more bigger, like this trace in the calcium, like the channel opening. Like we have this deformation on the cynetic or the response of the calcium, because sometimes the photophysics or even the properties of the fluorophore are like shaping more this type of curve, like even the natural phenomena. And I would like to know, like, if you're contending with this kind of phenomena, or it's just irrelevant because of the temporal scales you're extracting.
So we use lots of different ways of releasing calcium. Obviously, if you're doing it IP3 dependent, IP3 independent, you get these different transients. We find quite different signals, right, when you release the caged IP3, especially if you're saturating it. But we find quite similar responses in terms of, like, with the ACH and the IP3, caged IP3. I wouldn't say we get, like, vastly different, averages at the end, but I'll have a think about it and come back to you.
Thank you. Thanks so much.
Okay, thanks very much.
Now, please welcome Moara Lemos from CNPEM, Universidade Federal do Rio de Janeiro.
Hello everyone, I'm Moara. I'm a professor at the CNPEM that Emiliano just introduced a few minutes ago. And today I'm going to talk about the sample preparation for cryo-electron tomography and lamella preparation under the perspective of flagellar structure in Trypanosoma cruzi by cryo-electron tomography. So, Trypanosoma cruzi is the etiological agent of the Chagas disease, a disease that affects more than 7 million of people, mainly in Latin America. This protozoan parasite poses a single flagellum, which is persistent along of the cell cycle and has an impact on the cell motility and cell morphogenesis. The flagellum, it's a complex structure formed by the axoneme, which is composed by 9 microtubule doublets and the central pair, and by an extra axonal structure named the paraflagellar rod. So we are interested, our group is interested in to study the intraflagellar transport, which is a bidirectional movement of flagellar proteins between in the matrix of the flagellum. So this transport is driven in the anterograde direction, bringing proteins from the cytoplasm to the chip, and this is driven by the kinesin. And in the chip, the cargo is made free, and then the dynein recycle the proteins that are in the chip from the cytoplasm. And then we can see these IFT trains, what is called this arrangement of proteins, as electron densities along in the side of the flagellum close to the specific doublets. Then it was the first observed in Trypanosoma cruzi by our group. Oops. Then we used the cryo-electron tomography strategies to study, to have a deep look into these intraflagellar transports. So we used two different strategies. So one was the lamella preparation to reach a deeper region of the cell body of the parasites. And then we could see this region, which is the base of the flagellum, where we can see these intraflagellar trains being formed as a small arrangements in zigzag in here showed by the arrows. And then along the flagellum between the axoneme and the flagellar membrane, we see longer trains sandwiched between these, the flagellar membrane and the axoneme. So when we look directly in the flagellum, because the thickness of the flagellum is thin enough to be directly observed under the microscope, we could see novel circular structures in the Trypanosoma cruzi flagellum, which is also located between the flagellar membrane and the axoneme. Those are organized as few structures pulled together, or in many circular structures forming like a big group, forming some kind of protrusions between the membrane of the flagellum. We also observed those thin filaments, very similar to actin filaments, observed in the tip of the flagellum. So our plans, those are our initial data coming from the recent microscope that we have, the Titan cryos, and then we want to look to the molecular identity of those structures and compare them with the IFT trains. So if you're interested in cryo-electron tomography, sample preparation, trypanosomes, or intraflagellar transport, come and visit my poster, the number 30. So thank you very much.
And now the next speaker is Laura Estrada, the host from Universidade de Buenos Aires.
Good, thank you. Okay, so first of all, I would like to thank LABI committee for for organizing this amazing workshop. It's always nice to have the opportunity to share with experiences with so many friends, colleagues, and students. So thank you, Oliver, Leah, and the entire team. So I'm Laura Estrada, I'm professor here. I'm the director of the biophotonic group at the Quantum Electronic Laboratory at the School of Science. Our lab is right crossing the street, maybe in that direction, that direction, okay, good. So it's working, no? Okay, so in my group, we use infrared pulse lasers as tools to address biological questions. And the main interest of our group is we can divide it into two main categories. One is to study intracellular dynamics, protein transport, protein-protein interaction, protein-membrane interaction, protein oligomerization. And on the other hand, we use infrared lasers as a tool to fabricate optical fabrication of micro devices. So on the side of biological intracellular dynamics, we perform, do we have a laser here? Yes. We perform single particle tracking, so we can track proteins and molecules once at a time. And we also do 3D reconstruction of moving fibers inside living cells by orbital modulation. And we also perform and applied a lot of fluorescence correlation spectroscopy techniques from the very basic point FCS to more advanced fluorescent FCS methods. And we develop also new application of those methods, most of them in collaboration with Enrico Graton and his team during the last years. And on the side of optical microfabrications, we have developed a method and using photosensitive resins along with 2-photon excitation lasers to fabricate rapid and low-cost micro devices. So this is our very first microstructure. It's a micro messy playing football here. And from this very first microstructure, we now can fabricate 2D, 3D, and even fluorescent microstructures within minutes and, I don't know, maybe $1 or $2 cost. So of course, all these projects are highly interdisciplinary. So you can see here my local current collaborators. They are all from different institutions. Okay, so they are all from different institutions, but they are all from biology departments. So I'm a physicist, and so this is really interesting. So most of my actual collaborations are here in this same building. And in order to address these questions, we develop specific instrumentation. So this is how our homemade instruments looks like. So it's very different compared to a commercial microscope. So this is a 2-photon orbital tracking microscope. So this is not working, but okay. And we also develop our own software to acquire data and to analyze data. And regarding sample preparation, we have access to cell culture rooms and also clean rooms. So this is interesting because this is Natalia, Mariana. Mariana is a physicist, and he's Francisco, who is also a physicist. So we are able, with help of a lot of people, some of them are here, to do some wet lab biology work also. And finally, so this is the people, the students who joined my lab since I returned to Argentina, like from my postdoc with Enrico Graton, like 10 years ago, and they have done almost all the work in the poster. Some of them are still around, so you will probably be able to see Ignacio and Joaquin, Maria Jose and Constanza, Nico and Mariana, and I mean, please stop by our poster, and this is a nice opportunity to, it's an excuse, let's say, it's an excuse to chat, to discuss about these projects and other things you want to discuss with us. So please stop by, and probably you can also, we can also share a mate.
Thank you so much. Now I'm glad to welcome Greg Kitten from Universidade Federal de Minas Gerais.
Okay, so I'm going to tell you about the spectroscopy SIMS network that we're building. I want to tell you a little background quickly. So I'm from Minas Gerais, Belo Horizonte, UFMG. You can see we have, so we have somewhere marked here, you can see the arrow. And this is where the center is located. We have a very specially, a building that's rather large. So we have 3 transmission scopes and 7 or 8 working scanning electron microscopes, all with different detectors. And we do lots of teaching and classes also in the center. You can see a couple of the microscopes here. But the point I want to go, and this is when I want to ask this question, what is your dream microscope? So Lionel has hundreds of dreams, sometimes nightmares. So I think all of us have gone through this phase where you've been around a couple of years and you all have the microscope that you want. And so most of those will be something where you have resolution in the nanometer range so that you can study cellular architecture at a high level and detect changes when you compare diseases and other methods. So that's a pretty good basic goal. But then there are also, you want something that you can directly detect and identify material that's present, proteins, lipids, carbohydrates. We've been hearing about, we're limited by antibodies that we have that don't work. And so most of you have probably done SDS page gels, so you know how many bands you have on a gel that are unknown. And so you sit there like, okay, what are these things that are important? And so these things have been running around in my mind. I have a lot of developmental biology questions that I still haven't been able to approach until now. So I retire next year, then I'm going back to the lab to work on those problems. So one of the first things I want to say is that I have two technologies, and I've wanted these for years, and now we have them. It's not a microscope. The first one is that you can see here is one of the volume EM techniques. Not spend too much time here. So you collect hundreds or thousands of sections in Harvard and I forget the institute in Seattle. They've collected over 6,000 serial sections, mounted those, and the connectome is all built on that basis. The thing that I want to remind you, if you look at like, this is one of our first, Mario is sitting here, he has a poster and he'll talk more about this. We have next year have a symposium or workshop over this. But the point I want to make here is that when you get this data, this is still in the initial phases of it. You can collect all this data and you get great images, but the big thing for me is you can do volumetric analysis. How much volume, how much area do you have for mitochondria? What does that change in Alzheimer's, in Parkinson's? So you can start doing, with any tissue you pick, so you can start looking at all sorts of morphometric data. Again, Mario has a poster over this, we can discuss more. The second point, the thing that I've always wanted for years was some way to do mass spectrometry imaging, MSI. And so you can see, most of you, I'm presuming, will know most of these. So there are a couple of different ways that you ablate. You get ionization, material goes off, and then you measure it. And so those different methods of mass spectroscopy, now those are imaging. You can also image a tissue, but it's not very good. The resolution is not good, and you're never quite exactly knowing if you're in the right place. So now with these new systems, the newest systems that are coming, the secondary ion mass spectroscopy, they can't go into the way they do this, but they have now for biology new types of cluster beams that you can ablate, and you can get peptides and lipids and carbohydrates fairly intact, at least up to 20,000 Daltons. So let me jump here. So this is the second part, is that all of this came about a lot. We got a grant, but we didn't know anything about it. So then because of CZI, I made a contact with this fellow, Sebastian Van Uffel, started talking to him. I won't go into details here, but then we met a Brazilian that's working in a very good lab in the UK, NPL, and he invited us to a workshop that happened to be in Brazil. So then we go to the workshop and we run into other people, and this is a fantastic group to be in a workshop, so we learned everything we did. We bought an instrument, I'll show you, but also there I met Lara Baque from Argentina. So started discussing with her the possibility of setting up a SIMS network. So now we've got this number of people, a lot of people scattered over the place. I don't have time to talk too much about any of the instruments they have. So we have a very good group and it's building. We have other people that are coming in from Europe. They want to really help us build this network. And so it will be our instrument will arrive in December and then probably be mounted in early next year. This is our instrument. So it's an Ion Optica. It didn't fit in our room, so we have to actually remodel the instrument because we can't change our room. Room is 6 meters by 5 meters and it doesn't fit. So I, because of time, don't have time to go through this, but you can do also mass spec, and then you have all the peaks that you got. You can do this, but you can put all this together and you can get imaging to do in 3D. You can do cutting down and do 3D imaging with this mass spectrometer. These, again, we have a huge AI part of this project that's being constructed. We've already got people from our computer department are very interested, and also Google probably will help us. This is like, I just want to mention one or two little quick points here. So usually you have the multiplex that you do with all of these. This is in a case of 15 different multiplex. But when you use this TOF SIMS, there's a way, and people are using nanobodies, primary nanobodies, labeled with lanthanides. And so you can do up to, counting lanthanides and other metals, you can get 54 different biomarkers labeled with primary antibodies. Then you throw them all on at the same time, and you do one analysis, and you interpret the peaks. So instead of spending weeks, you spend one morning. And then you run the analysis, and so this is all coming out. So this is a professor from UFMG. She got one of the I4A grants that was mentioned earlier, so she's now spending a year in Sebastian's lab in Maastricht. So another connection that went further, and so now she's working on developing this and also analyzing all of the carbohydrates, and she has contact with INCA, the Brazilian Cancer Research Center. And I'll stop there. So anybody has questions?
The speakers also have a poster, so we can make questions and comments directly. Okay? Now the next speaker is Thayna Avelino from CNPEM.
Hello, everyone. Thank you, Labi, for the invitation. Thank you, organizers, for the kindly invitation. Today I will talk a little bit about our experience in CNPEM with bioimaging. My focus will in optical microscopy. My name is Thayna Avelino. I'm a bioimaging lead in CNPEM, specifically in LNBio. CNPEM is a national research center located in Campinas, São Paulo, Brazil. CNPEM houses 4 national laboratories. One of them is devoted to nanotechnology, bioenergy, the Synchrotron Light Source Laboratory, and the last one, Bioscience Laboratory, where we work with optical microscopy. In my work at LNBio, we work to provide free of charge training and access to advanced optical microscopy. We receive around 115 per year of users. Mostly of them coming from Brazil, but we can receive users from across Latin America and the Caribbean. Monthly, we can offer grants for these users to come do their experiments and receive training in optical microscopy. To know more, please visit my poster, number 39. A part of our work to spread the word of optical microscopy, we are working to developing methods across the scales. We are working with images. Oh, sorry. Spoiler. We are working with images from entire animals, from super resolutions. And we are trying to develop pipelines to correlate bioimaging from light microscopy to synchrotron imaging. We have a lot of beam lines in CNPEM or synchrotron light source. So we are trying to combine all of our imaging modalities to resolve biological problems. To know more about this beautiful work, please visit the Umberto's poster. Thank you, Labi. Thank you, all my friends, my group, and LNBio for all the support.
And now, Armando Castillo will talk about bioimaging in Panama.
Thank you. Sorry for the delay. My name is Armando Castillo from Panama. It's a pleasure to be here today. I'm going to explain a little bit of the story of the Center for Bioimaging in my institute. So, in the past, there was nothing. All the bioimaging capacities in Panama were located at Smithsonian Institute. As you know, Smithsonian Institute is an international mission located in Panama that do a lot of research there. The problem is that at any given moment, Smithsonian could deny us the use of their equipment. So basically, all the ecosystem of research in Panama depended from these capacities from Smithsonian. And this actually happened this year when they basically closed the bioimaging capacities, and also they had the shutdown, so we cannot access those resources. But we knew that from the past, so we basically applied for money from the Banco Interamericano de Desarrollo and acquired 2 new high-end equipment for bioimaging, this ESEM and this confocal microscope. And so we basically created our new bioimaging system in DICASAT. So we have these 2 equipment. We are expanding for more equipment in Panama, and we give services to the whole research community in Panama now. So we actually work with the fabricantes. We have this core facility. We give training to people in Panama, all sort of microscopy. We also produce our own in-house protocols, and we work very close with the researchers to give the best services we can. So one of the things we do is now we absorb all the capacities for bioimaging in Panama. So even Smithsonian is using our research centers now because they closed theirs. And we're basically working alongside all these different institutes in Panama giving services for bioimaging. So we now have like 18 different projects, 8 different research groups working with a confocal microscope, and a lot of research going on with, especially with biomaterials and semiconductors from different universities in Panama. So we joined LABI last year, thanks for a visit, surprise visit from Oliver. I think you came to Panama and say, hey, why you don't join? We are now part of Latin American Bioimaging. And this is the complex we work here. This is my institute in DICASAT here. And we are building a new building here. And we are buying new equipment. And one of those is high content, high throughput microscopy. We're also working with one of these 3. We have open convocatory for buying one of these equipment. We want to do a lot of research in cancer, and we are buying a cluster of supercomputing to work with AI, doing imaging as well in Panama. We already have different theses. We are about to publish this. Also, this is a student from Spain who came to Panama to actually do this with us. And we do a lot of research, publish outreach. So we push really hard with the media around so people can see the work we are doing in Panama. So we have a Instagram page. Please follow us. And I'm going to show you really quick the work I do in Panama. I'm actually a neurophysiologist. I work with ants, and what I want to do is to work with ants and the evolution of their brains. So this is an antenna of an ant, and we actually study the brains of the ants and correlate the different sections of those brains with the sensory input. And with the students, we reconstruct these brains. We look at the different sections of these brains, and we compare a lot of species of ants to see how the brain evolves. So this is another ant. This is a nocturnal bee. This is a wasp. And one of the techniques we are trying to develop is how can we get through the cuticle without dissecting the brain. Easy. My student pretty much make them invisible. And we can now scan the whole brain inside without breaking the head. Really quick, then we reconstruct the whole thing, and we can do now whole months of the insects in one go. And we are applying this to other insects. This is an ant. We can see the whole muscles, all the structures, the brain, the glands, and without micro CT. And we are about to publish this as well. And we can apply the same technique to different insects. This is a mosquito here. and we can do all sorts of reconstructions in the brain. So if anyone wants to work with me on this kind of research, insects, small animals, we can do that here in Panama. And you are all invited to come to Panama to collaborate. And so some images we will get there. The team and the students.
Thank you, Armando. So, Oliver, do you have some announcements or?
Yes,
Okay, so thank you all. Let's networking, guys.
So thank you very much, all of the lightning talks. Really great job. So we invite you all to visit those posters and those work and talk also with the people.